I want to welcome everybody to the fourth PKD RRC Annual Symposium. First, I'm just going to remind you what we're here to do. We develop and share resources, reagents, and expertise to advance innovative research in the field of polycystic kidney disease. Today's symposium objectives are listed here. First, we want to promote broad awareness of advances in PKD research, and we want to highlight existing PKD research resources. Part of our mission is to identify state-of-the-art techniques that could be applied to advance our understanding of PKD biology. We want to foster interaction among members of the scientific community. We hope to discuss challenges and gaps in PKD research that, if we address them, could move the field forward. And then we want to highlight the tools and resources developed by the PKD RRC. And so here's our overview of day one. For those of you that didn't have a chance to look at the agenda, we're going to start off by hearing about the patient experience, which is why we're here in the first place. We're going to have a session on GLIS2, which is a new player in polycystin signaling. We're going to hear about data discovery science applied broadly to the kidney and to maybe get some hints about what we might do. We're going to have a lunch break at around sometime afternoon. And then right after lunch, we're going to talk about the PKD RRC. This is our last year of funding for this current cycle, and we want to sort of lay out the progress that we've made. We're going to talk about clinical advances, and then just to remind everyone that trainee and early stage investigator abstracts are kind of sprinkled throughout the program. Housekeeping important. Everybody is going to be muted with no cameras except for the speakers, the session chairs, and panelists. It's because we have a lot of people registered. We're on a tight schedule. So for speakers, no hard feelings. We're going to give you a 5-minute warning, and we're going to try to end all talks on time. All questions are going to be handled through the chat. So if you have a question, submit your questions and comments to everyone. If you have a technical issue, please contact John Hare, who's running the meeting, and Nicole Reed here at Maryland through the chat. Just to remind you, after this symposium, we're going to distribute an evaluation survey, and please do fill it out because every year we make adjustments based on what you tell us. I want to thank all of the speakers, the moderators, and session chairs, and I want to give a special thanks and shout out to Nicole, who has been instrumental in helping organize this, to Patricia Uteda and Pam Tran, who were the chair and co-chairs of our organizing committee. And of course, as always, support for the PKD RRC and this symposium comes from NIDDK, NIH, and we appreciate their support. Just want to remind everyone, we do a lot of stuff. Please get involved. Visit our website, keep abreast of the pilot project programs. We have monthly scientific roundtables. We're on Blue Sky, so please follow us. We have a mailing list and a discussion board. And then finally, we can never get enough of PKD research. So don't forget to register for the PKD FASEB meeting, which will be June 29th to July 3rd this year in Southbridge, Massachusetts. And so with that, I'm going to turn it over to Susan Gleason. The title of her talk is The Patient Experience, Our Stories, and Your Impact. And just by way of introduction, Susan is a member of the PKD RRC Patient Engagement Group, and she's the first person in her family who was diagnosed with PKD, having been diagnosed and in a workup for a surgery. She's a retired attorney. She graduated from Northwestern University Pritzker School of Law. She worked first in private practice, and then she served as a law clerk for the federal district courts in Illinois for over 20 years. And with that, I'm going to turn it over to Susan. And I will stop sharing.
Thank you. I'm so excited to be with you all here today to talk a little bit about the patient engagement group at the RRC. As you know, my name's Susan Gleason, and I've been a member of the PEG since its inception in 2023. When I was diagnosed with PKD in 2008, it was incident to my third hernia surgery. It was a shock. Never had it in my family, never had any kidney problems in my family at all. I think my husband and I spent a couple months in denial until the genetic results came back showing that, yes, I had the PKD1 mutation, which was described to me at that time as definitely pathogenic. Ever since then, I've done everything I can to educate myself about PKD with a particular interest in the research into curing PKD for myself, but also for my two sons and my granddaughter, and hopefully future grandchildren on the way. Because of my interest in research, I was naturally drawn to the RRC. The incredible leadership of the PEG facilitators has what made me stay here. I just want to thank Dr. Mustafa, Dr. Gay Woodford, Veena Rao, PhD candidate, and project manager Nicole Reed. Without them, this group wouldn't be possible, and they've really made this experience a wonderful one for me. Our PEG members are scattered across the country, across time zones, which can make meeting challenging. You're going to hear later from Julie Hogan, I think it's 6:30 a.m. Where she is right now. We have a broad range of work experience. Some of us have scientific training. Others work with transplant patients and PKD patients to help them with those complex processes. As you already know, I'm a retired attorney from Springfield, Illinois. We have a wide range of disease experience. Two are relatively asymptomatic, including myself, despite my mutation. One has stage 3. Two have significant to severe polycystic liver disease, in addition to the PKD, and 3 are post-kidney transplant. Our mission as put to us is to find a way to include the patient perspective into the decisions about the resources developed and to be a vital part of advancing PKD research. And that's a wonderful mission, but exactly how are we going to go about this? That is the challenge we faced as the first patient engagement group in the RRC. Our approach has evolved, and I will continue to evolve with the organization. One way we decided to engage is to have a PEG member attend and actively participate in subcommittee meetings and then report back to the other PEG members. In other words, act as a liaison between the committee and the PEG. There are currently 5 committees, if you don't know: the steering committee, in vivo PKD models and reagents, cell models, antibodies and vectors, and clinical resources and materials. We decided to start with just 2 committees and see how that went. And after a couple of months of that, we realized we need a much clearer idea of what we're supposed to be doing at these meetings and what we're supposed to be doing at the PEG meetings. And this led to the development by the facilitators, with input from us, of a document that clearly spells out what is expected of us. I want to highlight just some of the major points of that governing document so you can get a sense of how this works. In general, each of us commits to attend a monthly subcommittee and monthly PEG meeting and to be responsive to requests for additional input from wherever those requests come. We're encouraged to get together socially outside of our official duties to get to know each other better. I found that the total commitment's about 3 to 4 hours a month, sometimes more, sometimes less, depending on what's going on at the time. As far as subcommittee participation, we're there to represent the range of patient perspectives, not just our own. We're responsible for learning what the subcommittee does and asking questions when we don't understand, and relaying information between the committee and the PEG. and also educating the PEG about the subcommittee. What are some concrete examples? I can speak to the steering committee, which I've been attending with my fellow PEG member, Sarit. I get a bird's eye view of the organization. And I was thinking back, I listed just a few of the topics that have come up in those meetings. And I remember being particularly interested in the review of grant proposals and grant applications because of my interest in the research. This list doesn't mean I've offered a patient perspective on each issue or even on most of these issues. Not every topic lends itself to a patient perspective, and most of the time I still spend listening, learning, and asking questions. But some topics for patient perspective can be relevant, such as social media presence, review of websites, etc. And I have tried to offer my perspective on the issues, on those kinds of issues. I think knowing when a patient perspective is useful is a skill I've gotten better at and will continue to improve as my participation continues. The other committee with current PEG representation is the Clinical Biomaterials Committee. PEG member Julie Hogan attends those meetings, so hopefully I'll be able to pass it off to her. Hopefully she's here.
I'm here and I'm ready, Susan. Thanks.
Oh, okay.
Hi, I'm Julie Hogan, and like Susan, I don't have a family history of PKD, and more recent genetic testing before my kidney transplant revealed a pathogenic variant. I did receive a pediatric en bloc kidney transplant, which is 2 little kidneys donated by the family of a baby who had passed away. And thanks to their generosity, I never had to do dialysis, and I just celebrated the 5th anniversary of my transplant recently. And I currently work to help patients get listed and find a living kidney donor. I've been participating in the clinical committee for the last year or so, and the committee's been working tirelessly to harmonize the data from 3 institutions into the PKD RRC joint database to make that data available to all of you and anybody else who's interested. This is a living database with ongoing addition of information, and it's now available as sort of a one-stop shop. So when you put in a data request, you just have to do one to get the information from all 3 institutions to make it easier. You'll hear more from Dr. Seliger later in the symposium about the database, but we hope you'll take a look at it and what's available on the website and take advantage of this resource. I've been incredibly impressed with the level of concern and care taken to ensure that the patient data is protected and can't be connected back to the research participants. Sometimes I've even felt like it's in an effort to be incredibly thorough, the amount of time and energy spent on this might be more than is required. And I voiced some of my thoughts on this. And really the reason is to encourage the urgency that patients feel to move the research along quickly. The group was recently asked for the patient perspective on sharing some more specific genetic information on the general class of the genetic variations and the impact of those variants, like the disease-causing element, and into the database. This data was not originally added to the database. However, it became clear that it was important information that researchers were looking for and wanting. So I brought the question back to the patient engagement group and explained the background and for why we would want this more specific information added. And again, wanting to also protect the patients and families who had agreed to participate in this research. The patient engagement group spent time reviewing the consents the participants and the parents had signed. And we all felt confident that those signing the consents would feel very comfortable with their, and had the understanding that their information would be used in this way, not only by the institution who gathered the information, but putting it into the joint database would allow for more easy access. And the efficiency of that is much improved over, researchers having to go to 3 different institutions. And that makes a lot of sense to us as patients feeling that urgency to find answers and treatments and hopefully a cure. Some of the PEG members, including myself, have participated in genetic research like this, and we've signed consents like this. And from our perspective, sort of not only would we hope that our information would be used, but we would expect it to be used. And leaving it out or not using it, would actually sort of disregard what our preferences are as far as we want this data to be used. It's kind of a hassle to participate in research sometimes. And so the extra effort that we're taking to, provide this information and come to meetings and do the things that we need to do to participate, we really want our information used. So if we're signing those consents, we want our data to be used as much as possible in an effort to find the treatments and cures. We also were asked to write up a statement documenting our thoughts on this, and now that's part of the information that's available through the patient engagement group and through this subcommittee. It was also suggested that we write a patient voice article on this topic, and we've actually started working on that. So I've learned an incredible amount being part of this committee. I find everybody who participates to be kind and receptive and interested in the patient experiences and perspectives. And the members of the group really hope to become more involved in more of the committees and other projects like this in the coming months and years. And we encourage you to reach out to us with any topics where hearing the patient's perspective might be helpful. I will hand it back to Susan.
Thank you very much, Julie. Other opportunities we've had for development include the monthly science roundtables. For myself, I've attended almost every one of these. As you probably know, they're presentations given by experts for experts in the field. Listening to these presentations has greatly increased my knowledge of PKD, which I hope will eventually help me become a more effective PEG member. If you haven't yet attended one of these, it's a wonderful opportunity to discuss PKD research at the highest level. I think the next one is April 7th. This symposium is obviously a great way for us to reach out to PKD researchers. I can't think of anywhere else that I can share my story with so many PKD researchers. You never know, one of you might hear it and think of a new avenue of research to pursue. I also want to highlight a white paper that is being written by PhD candidate Veena Rao. She's been a fabulous facilitator of our group. This white paper is being prepared for publication in a patient research center journal. And for this white paper, we were all asked to complete surveys and contribute our comments on the various drafts over several months. It was really a great learning experience for me, and it's exciting to think that I might play some small part in expanding research on incorporating patient perspectives into research. What are our goals for this year? Well, we've got 3 more committees that we need to staff with PEG representation. The facilitators have recruited new members, and we hope to be onboarding them soon for this purpose. We hope to continue recruiting new members because our ultimate goal is to have 2 members staffed for each subcommittee in order to cover absences and spread the workload. We also want to develop what we call a boot camp, which are presentations about each resource, what the resource is, how it's used in studies, what published studies there are. Has it had any effect on advancing PKD research? Because we think that understanding each resource better will help us give useful input at these subcommittee meetings. What can we do for you? Well, Julie touched on that. We can provide a patient perspective on your proposed studies, and we can also help with any text that needs to be understood by people who aren't scientists or physicians, like abstracts, patient consents, websites, and the like. We also, as you heard, can share our stories, which might just give someone researching this a disease a new idea or a broader understanding of the disease. I'd like to end by saying how thankful all of us are for the RRC. We've been amazed at the tireless dedication of everyone in this organization. Your work is incredibly meaningful to patients, and we know it does make a difference. As patients managing this difficult disease, your work and the work of every PKD researcher gives us hope for ourselves that one day there will be a cure. Again, thank you, and please let us know if we can help you in any way.
So we'll turn it over to our moderators. Thank you, Susan, and Julie, that was really moving. Thank you so much. Let's turn it over to our moderators for the next session is Doreen Peters and Yanjun Jung.
Thank you. This was informative and impressive. But we now continue with the scientific session, session 1 on GLIS2. GLIS2, a player in polycystin signaling and PKD therapy. And I'm very happy to introduce to you the first speaker, who is Dr. Anton Jetten. Anton Jetten, he is Deputy Chief of the Immunology, Inflammation and Disease Laboratory and Head of the Cell Biology Section at the National Institute of Environmental Health Sciences at NIH. His research has been focusing on the study of the GLIS1, 2, 3 transcription factors, which actually have been discovered by his lab. So the title of his presentation will be Roles of GLIS2 and 3 in Normal Kidney and Cystic Disease. And Anton, please, the floor is yours.
Well, thank you very much for inviting me to this, to give a talk about GLIS2 and GLIS3. So as was just was said, that the GLIS family was identified by a postdoc in my group, and we discovered 3 members of what we call the GLIS family of Kruper-like zinc finger transcription factors. The name comes from that the proteins were very had high homology with the GLI proteins of transcription factors. And the GLI proteins are very important in development and cancer, and are transcription factors that act in the hedgehog signaling pathway. And these proteins have an highly conserved DNA binding domain that consists of 5 zinc finger motifs. And the zinc finger motifs, they recognize an DNA element that we call GLIS binding site, or GLIS-BS, that consists of an highly G-rich region. The proteins are, except for the DNA binding domain, they're highly conserved, but outside the DNA binding domain, the conservation is very little, except for what we call an highly conserved region that GLIS3 shares with all the members of the GLI family. And here, here below is shown the sequence of the homology. And there is particularly in certain region that is called ciliary localization signal that has been identified and described by others that are studying the GLI protein family. And that sequence recognizes a protein that's called transportin that can actually bind to that region and import the protein into the primary cilia. And the other proteins, GLIS1 and GLIS2, don't have this highly conserved region. So this indicated that GLIS3 may also be localized to the primary cilia. And that is indicated here by immunohistochemistry. Here's the primary cilia identified by staining with alpha-acetylated tubulin. And you can see here is the staining for GLIS3, and GLIS3 localizes at the tip of the primary cilia. And that's very similar what has been identified by the GLI proteins. And Naturi, he has done proteomics on primary cilia, and he actually identified also the GLIS protein as part of the primary cilia. So there's several types of evidence that GLIS3 is localized to the primary cilia. So this is kind of our model. We think that GLIS3 may act via two different signaling pathways. One that uses this ciliary localization signal that imports GLIS3 into the primary cilia. And then we hypothesize that there is probably some kind of signal that acts via an G protein coupled receptor that modifies GLIS3 activity in some way, and then localizes GLIS3 to the nucleus where it regulates gene transcription. But it not always works this way. Sometimes there is also, and we have evidence for that in the thyroid hormone system, that there is a G protein coupled receptor at the basal membrane that then regulates GLIS3 activity, and then via that mode regulates gene transcription. As I mentioned, GLIS3 plays a very important role in a number of different diseases. I'm not going in detail into this, but everything that's here listed in blue are either very large deletions or point mutations that result in dysfunction of GLIS3 activity. And this leads to, in patients and also in mice, the homology of the phenotype between humans and mice is very similar. And that leads to a kind of multi-organ phenotype that consists of neonatal diabetes, congenital hypothyroidism, and also polycystic kidney disease. And that's what I'm going to focus on first. So the loss of GLIS3 function results in an autosomal recessive type of PKD. And cysts are being formed in various parts of the nephron, and that leads to, in the terms of progression, participates in fibrosis, inflammation, leads to end-stage renal disease. And you can see here a histology of the GLIS3 deficient kidneys. In the case of the GLIS2 deficiency, that leads to a different type of cystic renal disease called nephronoptosis, and you're all familiar with that. And also there you get kidney atrophy, formation of cysts, and also here fibrosis and inflammation plays a very important role in the progression. So our goal is that since the GLIS proteins function as transcription factors, we are particularly interested in what genes are they regulated. And what does that tell us about the biological functions of these proteins in the kidney? And also, can we distill from this the causal mechanism by which these proteins lead to disease? And eventually, we hope that it also will lead to therapeutic strategies. And we use a number of different strategies to look at that. Molecular strategies like RNA-seq, single nuclear RNA-seq, ChIP-seq, and as well as proteomics and metabolomics and spatial transcriptomics. As you know, that just after birth, the kidney undergoes still a lot of different changes, and that's well described in the literature. And that includes a shift from aerobic glycolysis to oxidative phosphorylation. But also an increase in the large number of transporters. So a lot of the epithelial cells in the nephron become more mature and get a better function. And also a lot of changes in lipid metabolism. And that's, for example, indicated here. We're concentrating here on mitocarta genes, but are basically genes that are related to mitochondrial functions. And it's about 1,500 genes. And you can see when we compare in the mouse, PND28 versus PND14, you can see a large number of these mitochondrial related genes are being induced in normal kidney. So to understand what the differences are between gene expression between wild type and GLIS3 knockout mice, we did bulk RNA-seq. and pathway analysis of differentially expressed genes identified a large number of transport genes, as well as mitochondrial genes. Aerobic respiration was also mitochondrial related, and then ion transport related genes. The genes that are upregulated are related to inflammatory responses and environment and extracellular matrix related genes. If we look at the mitocarta genes, how they are regulated in GLIS3 knockout mice, you can see most of the genes are suppressed in GLIS3 knockout mice. So it's the opposite. The genes that are upregulated in normal kidney are all suppressed in the GLIS3 knockout kidney. And so that what this tells us is that the knockout of GLIS3, of deficiency in GLIS3, leads to metabolic reprogramming, and this renal cells are more reliant on aerobic glycolysis instead of oxidative phosphorylation. In addition, that the inhibition, the suppression of transporters indicates that the renal cells are not as mature, and in the GLIS3 deficiency inhibits kidney maturation. That's further illustrated here. There's a large number of mitochondrial metabolism-related genes, and you can see here they're all downregulated in GLIS3 knockout kidney. If you look at cell proliferation genes, you can see they're all upregulated, and that fits with the increased proliferation and cyst formation in the kidney. And you can see here a large number of transporters, and in different segments of the nephron, they are all suppressed in GLIS3 deficient mice. One of the speakers are going to talk about the GPRC5A, and I happened to look at that, and that's greatly upregulated, and that was described as being a marker for the cyst in the kidney. We were then interested in what of these genes that are differentially regulated are directly regulated by GLIS3. So we did ChIP-seq analysis, and you can see here, one of the sites that come out is a consensus binding site for GLIS3, as well as other GLIS proteins. And it's also adjacent to that, we find binding sites for HNF1B, the Hippo signaling pathway, transcription factor TEAD, and NRF1. This indicates that these other transcription factors are likely playing also a role in the regulation of the genes that are regulated by GLIS3. And we particularly focused on HNF1B because HNF1B phenotype in deficient mice and in humans is very similar to that of GLIS3. So we were interested in what is the interaction between these two. So we did GLIS3 and HNF1B ChIP-seq analysis, and this graph here on the left side shows that there is a huge overlap between the binding sites of GLIS3 and HNF1B, two regions that are very similar to each other. And if we take the pathway analysis of differentially expressed genes that bind both GLIS3 as well as HNF1B to the same region, then we see that a lot of the genes that are associated with these pathways, like transport genes, epithelial maturation, what indicates that maturation of the renal epithelial cells is inhibited, while positive regulation of cell proliferation and ECM-related genes are all upregulated. So it really fits the phenotype that we see in the GLIS3 deficient mice. Here's an example of the genome browser tracks. You can see here the two mitochondrial-related genes. You can see here the GLIS3 binding, and it overlaps with the binding of HNF1B as well as NRF1. And what this indicates that these genes are likely that GLIS3 regulates these genes in coordination with HNF1B and NRF1. And you can see also here that two transporters. You can see GLIS3 and HNF1B are co-regulating several of these genes. And that indicates that GLIS3 for these genes functions as a transcriptional activator. However, in the case of the inflammatory genes, also here we see that GLIS3 and HNF1B can regulate these genes coordinately, but in this case, the GLIS3 functions as an transcriptional repressor. So GLIS3 can function both as a repressor, as activator of transcription, depending on the gene and the gene context. So what this indicates that in the healthy kidney, GLIS3 is needed for the normal transition from aerobic glycolysis to more reliance on oxidative phosphorylation. While if you're in GLIS3 deficient mice, when GLIS3 is absent, then this is disturbed. The oxidative phosphorylation genes, the mitochondrial genes are not being induced. And that's also kind of a Warburg effect. And that is an effect what leads to increased proliferation. So that indicates that GLIS3, and it confirms that the loss of GLIS3 leads to metabolic reprogramming and inhibition of kidney maturation. So in the last minutes of this presentation, I'm going to talk about the GLIS2 function. As I mentioned, it leads to nephronoptosis. And as you can see here, it shows the progression of the disease. You can see at 1 week, there is actually very little difference between wild type and knockout mice. And the biggest increase is between 2 weeks and 1 month. You can see a large number of differences occur between wild type and knockout mice. And what is interesting, if you look at here, these are all the genes that are upregulated. And these are the genes that are downregulated in blue. And the red genes that are upregulated are much more than the downregulated genes. And what this indicates is that GLIS3 largely functions as a repressor of genes in the kidney. When we look at the genes that are upregulated at 1 month and do pathway analysis, we see that a lot of inflammatory response genes and genes that are related to extracellular matrix are greatly upregulated in that in these kidneys. And for example, here you can see by qPCR analysis for 2 cytokines, you can see between 2 weeks and 1 month, you see a dramatic increase of these cytokines. So also here we were interested in identifying the genes that are directly regulated by GLIS2. And you can see again, GLIS2 binding sites are coming out together with several other binding sites. Again, the Hippo signaling pathway, and as well as HNF1B, and here this NR stands for nuclear receptors. Most of these binding sites for GLIS2 are in the proximal promoter region, about 33%. If we look at the GO pathway analysis and probably upregulated genes that are targets of GLIS2, then we can see a large number of the genes that are upregulated in GLIS2 deficient mice, about 60% are direct targets for GLIS2. And you can see again, inflammatory response genes related to extracellular matrix are the major pathways that come out. So GLIS2 is a very important regulator of inflammatory response and extracellular matrix related genes. And here is again showed the genome browser tracks, and here we compared the binding peaks for GLIS2, HNF1B, and we also compared it with GLIS3. And you can see for certain genes that are co-regulated by GLIS2 and HNF1B, but not regulated by GLIS3, but other genes, for example, the CCL17, are regulated by all 3 transcription factors. And for example, the TLR2, only by GLIS2 and HNF1B, while NMSTS4 is regulated by all 3. So, so that indicates that GLIS2 functions as a transcriptional repressor, and we have identified by mass spec analysis on a number of proteins that bind to GLIS2 that are forming a co-repressor complex and basically inhibiting the expression of a lot of different extracellular matrix and inflammatory genes. And that's shown further here that GLIS2 and GLIS3 can bind to the same binding site, the same GLIS-BS, and there is possibility for competition between the two. And GLIS3 functions as a co-activator, while GLIS2 functions as a repressor of gene transcription. So, and that probably plays an important role in physiology as well, this competition and the balance between GLIS2 and GLIS3 expression and how they regulate gene transcription. We were particularly interested in what's the function of GLIS2 in different populations of the nephron, of the renal cells in the nephron. And we know that GLIS2 is expressed in most of the renal cell types, epithelial cells in the nephron. And here, I don't have time to talk about all the different cell population. I'm going to focus on the proximal tubules. And as you know, 3 different types of proximal tubules have been identified as S1, S2, and S3. And here is shown the UMAP of the proximal tubules between wild type and GLIS2 knockout mice. And you can see here there is a population, what is called PT other, that is dramatically increased in GLIS2 knockout mice. And that's further illustrated here at the bottom, where you can see what PT other, you can see in the knockout is greatly increased, while the other population is not greatly different, except the PTS1 is a little bit lower in the knockout mice. So we're interested in what is the difference in these PT other cells. And what we found, there are a lot of inflammatory response genes, cells related to extracellular matrix, cell differentiation that are upregulated in these PT other cells. While the downregulated genes are genes that are related to transport.
5 minutes remaining.
Probably related to maturation. So what is indicated that these PT other is basically what has been identified as failed repair proximal tubule cells. And here is shown a list from a list of genes that have been associated as failed repair proximal tubule cells in several reports. And in yellow here is indicated the similarity between the marker genes for failed repair proximal tubules and the overlap with the genes that are in the PT others in the GLIS2 knockout mice. So that indicates that what we call PT other are indeed the failed repair type cells. So here's our hypothesis that in normal kidney, increased oxidative phosphorylation, what causes an increase in reactive oxygen species and DNA damage, basically injury of the DNA, as well as stress and injury conditions, injures the proximal tubule cells, and then you can have either failed repair or repair of the cells. And in normal conditions, the repair and providing healthy proximal tubule cells is basically the prevailing pathway, leading to regeneration of the renal epithelium. However, in the GLIS2 kidney, oxidative phosphorylation and all kinds of stress conditions also causes injury. But in the GLIS2 knockout mice, the failed repair pathway is prevailing, and therefore we see this increase in failed repair proximal tubule cells, leading to inflammation, fibrosis, and basically a senescence or SASP type phenotype. And that is responsible for the atrophy that we see in the kidney. Of course, you can ask me the question later. So there is really this balance of between failed repair and regeneration of the renal epithelium, and the GLIS2 seems to be playing a very critical role in maintaining this balance. So the loss of GLIS2 leads to increased inferior repair and less regeneration. So you can ask me the question in the question session, what happens if you overexpress GLIS2? And I can give you an answer on that. So I'd like to finish my talk by giving credit to all the people in my lab that have been working on the kidney project that are listed here in the pictures upstairs. And I want to particularly pay attention to Young-Sik Kim, what is the postdoc years ago, was in my lab and actually identified the GLIS1, 2, and 3 proteins in the lab. And here are all the previous postdocs that contributed to this work, as well as credit to several NIHS scores, and particularly Sarah Grimm, who has been very instrumental in analyzing all the RNA-seq, single-seq, and so analysis and as part of the bioinformatics core. I'm happy to answer any questions.
Thank you for this very beautiful overview of the GLIS2 and 3 proteins. So then, of course, first question, what happens when you overexpress GLIS2?
Can you tell me what you expect?
Can I tell what I expect? I expect that there will be more repair.
Yeah, and actually, so we have overexpressed GLIS2 in the kidney under an kidney-specific promoter, and what we see that these mice develop polycystic kidney disease very severely, and these mice only survive like a couple of days.
Yeah, of course, that makes sense. And is GLIS3 playing a very important role in this repair?
So,
because they are competitors?
So we actually, and there was a kind of a follow-up on, probably Stefan Somlo is going to talk about that in regards to PKD in the next talk. And based on his result, we actually also crossed GLIS2 knockout mice, this GLIS3 knockout mice, and we expected maybe to see a similar effect. And we see some reduction in the formation of cysts, but the cysts are still being formed. It's not as dramatic an effect as what was described for the PKD, where the loss of GLIS2 has a very dramatic effect on the cyst formation. But in the cross with GLIS2 and GLIS3, we don't see such a dramatic effect. There is some small decrease, particularly in the formation of large cysts, but it seems that the number of small cysts seems to be actually increasing.
Okay, thank you. I go to the chat. Feng Chen, is there any evidence that GLIS3 is similarly processed by cleavage within cilia for activation or inactivation as reported for the GLI proteins? The GLI proteins?
Well, that's a good question, and that's a very important part that we actually are pursuing, is what are the upstream signaling pathways that are regulating GLIS3. We know we have evidence, and we have published that as well, about a signaling pathway that is GPCR-driven, mediated in the thyroid gland, but that GPCR is not localized to the primary cilia. It's in the basal membrane. It activates PKA, and via PKA, GLIS3 is modified and enhances the transcriptional activity. So how GLIS3, when it's in the primary cilia, what we think is a GPCR-mediated signaling pathway, what the signal is, and what the pathway is that modifies GLIS3 activity, or maybe the transition to the nucleus, we don't know. And that's not something that's very easy to identify.
Thank you. The next question is whether there is a role of GLIS3 in non-ciliated and multiciliated cells, and have you compared its action to that of monociliated cells?
We actually have been looking at that. We know, for example, in the lung that the expression of GLIS3 is very high in the ciliated cells. And we are currently actually examining what the role is of GLIS3 in these motile ciliated cell types. And I think the role is very different, but we have to find out.
In your sequencing data, you mentioned an increase in repair markers specific to the proximal tubule. Do you see an increase of cell cycle regulation proteins? Is there a difference between cell cycle regulation in the PKD mutants compared to controls?
So, for example, is that with GLIS2 or 3? GLIS3?
That's not specified.
So for example, we have performed like EDU, what is an indicator of proliferation, and we see that the percentage of cells that incorporate EDU is increasing. In the GLIS3, the cysts are you have glomerular cysts, you have also cysts in the distal tubules and in the collecting ducts. Yeah. In the proximal tubules, they're basically dilated, but they form only very small cysts. The larger cysts are in the glomeruli, in the distal tubules, and in the collecting ducts. So, and in these cases, we see that if you look at the single nuclear RNA-seq data, there is where you see the genes that are related to cell proliferation increased.
What is the mechanistic role of GLIS2 in adult ADPKD?
Can you repeat that question?
What is the mechanistic role of GLIS2 in adult ADPKD?
Yeah, that's so we see, for example, in the GLIS3 mice, we see cyst formation prenatally. Yeah, so, and it has nothing to do, so when we look, all the results that we have looked at thus far is prenatal, and that's largely related, I think, to progression of the disease. So what happens, what is the very early effect? Yeah, when, and the initiating fact, of course, it has to be related to gene transcriptional regulation. As you have seen, if you look at PND7, the number of changes in gene expression is very low. And we have looked at these genes and see if it gives us any information about the initial cause, yeah, of these diseases. And we have not been able to identify a gene that could give us some clue about what the causal mechanism is of either nephronoptosis or polycystic kidney disease and GLIS3.
Thank you. This was the last question we can discuss now, but there are more questions in the chat. So thank you for your presentation, and I hand over to my co-chair.
Thank you. Now we are moving to the abstract presentation session. The next speaker is the Yoshiharu Muto. The title is a single nucleus multi-omics profiling of a mouse cystic kidney disease. Polycystic kidney disease identifies the GPRC5A as a conserved cyst lining cell marker. Dr. Muto, please.
Thank you for introduction. Today I'd like to talk about single cell multi-mix dataset we have recently published, and the GPRC5A as a cyst marker. To better characterize the mechanism of PKD progression, we generated single cell multi-mix dataset where RNA and chromatin accessibilities are simultaneously measured. On the late induction PKD1 deletion mouse model of slow onset PKD progression. And we collected the kidneys at P66, where nuclear cysts were observed, and at P100 and P130 with expanding cystic lesion. After sequencing and integration, we identified all the major cell types. First, we'd like to focus on distal nephron cells. We observed a large number, a lot of AQP2 positive large cysts in the medullary region in our PKD mouse model. And after subclustering analysis, we identified four subtypes with AQP2 expression. We annotated these cells subsets are cortical principal cells and medullary principal cells with other marker gene expressions. We have previously sequenced and analyzed human ADPKD kidneys and identified ADPKD specific cell state in collecting duct. And the gene expression signature of this cell type is closely associated with medullary or MPC1 cluster in mouse PKD kidneys. Gene set enrichment analysis indicated this MPC1 subtype enriched pathways associated with DNA damage response and energy metabolism activation. Furthermore, at later time point with a lot of cysts, this subtype enriched pro-fibrotic and pro-inflammatory gene expression signatures, indicating dynamic alteration or change in gene expression signature along time course in this cell type. Another interesting cell type in our data set was atypical proximal tubular cells. And the subclustering analysis, by the way, this cell type is like Dr. Jiten's data set, only detected in proximal in PKD mouse kidneys. And subclustering analysis identified three major subtypes. One of them highly expressed VCAM1, which is so-called failed repair proximal tubular cell marker, which emerge and expand after ischemia, reperfusion injury, or other kind of AKIs. And interestingly, the other two subtypes lack or are extremely low VCAM1 expression, rather show upregulation of CDH6, which is a development PT markers. And immunostaining analysis of the mouse model, our mouse model revealed the VCAM1 positive cells mainly localized in the cystic epithelia and expands or increase along PKD progression. And another interesting subtype, APT2, which lacks VCAM1 expression, is also PKD specific. Given the upregulation of CDH6, they may be regenerative PT cell states induced by PKD1 deletion or loss of polarity. By the way, immunostaining analysis also indicated even in the same cysts, some cells express VCAM1, some cells maintain LTL and seemingly normal epithelia, and others lack both of them, LTL staining and VCAM1 staining, in suggesting heterogeneity of PT cells even in the same cysts. To better characterize the pathways or differentiation path of these subtypes, we constructed pseudo-time trajectories, and this analysis indicated the VCAM1 positive APT1 subset and the other subsets are separately differentiated rather than on the same path. And motif enrichment analysis on chromatin accessibility or ataxic data suggested the VCAM1 positive APT1 cells enriched NFKB family transcription factor binding motif. And in contrast, the VCAM1 negative APT2 subtype enriched hippopathway effector TID family TID3 binding motif. Suggesting differential transcription factor activities regulates other cell states. We have previously identified GPRC5A as a cyst lining cell marker. Yeah, I'm really honorable. It's very honor for me to be mentioned by Dr. Jiten and Dr. Somo. So GPRC5A expression in mouse kidneys are mainly detected in principal cells, medullary, especially medullary principal cells, DTL, and atypical proximal tubular cells. Indeed, immunostaining analysis suggested GPRC5S are mainly localized in DBA passive collecting duct. GPRC5A also localized in this white LTL positive proximal tubular cells, as well as some of BKM1 positive cells also express GPRC5A protein. And we have also identified in our previous paper, the 5' distal enhancer, which is specifically activated in human ADPKD, was interacting with GPRC5A promoter and upregulates GPRC5A. We validated this enhancer by confirming targeting this enhancer down regulates GPRC5A expression in cyst.
2 minutes.
Okay, thanks. And interestingly, this sequence is conserved in mouse kidneys, and this enhancer is also specifically activated in PKD cell state. And this enhancer enriched TID family binding motif. Indeed, hippopathway deactivation by targeting LATS1 and 2 upregulated GPRC5A expression, suggesting hippopathway deactivation induced by PKD1 deletion may be partially involved in upregulation of GPRC5A. So what is the role of GPRC5A? In our data set, in our mouse model, upregulation of GPRC5A is associated with larger cysts at later time point. And this is not PKD, but interestingly, GPRC5A high expression is associated with poor prognosis of kidney cancer, as well as other many cancers, including pancreatic cancer and colon cancer. And we have performed RNA RNA-seq on GPRC5A knockdown on cystic cell line, and GPRC5A knockdown down-regulated many proliferation and inflammation associated genes, indicating GPRC5A may promote cyst growth by regulating proliferation and inflammation in ADPKD kidneys. To validate this hypothesis, we are generating GPRC5A knockout PKD mouse model to see GPRC5A is implicated in cyst growth in mouse model. And finally, I'd like to thank my postdoc mentor, Dr. Ben Humphries, and his lab members to help this project and support my transition to the independent position at the UT Southwestern Medical Center. And this project is mainly now being led being led by Dr. Seishi Aihara, new postdoc, and our new tech, Pretty. Thank you for kind attention.
Thank you, Dr. Gupta. So
Sure.
now we're going to take questions.
So the GPCR5 is also expressed in the distal the DTL. Do you see, so that means is the Hippo pathway more active in that part? And do you see kind of overlap?
Yeah, we also found GPRC5A protein expression is localized in DTL, so in normal kidneys, and yeah, Hippo pathway or also Hippo pathway effector TID as well as hypoxia upregulated GPRC5A expression in our hands and also other groups. So potentially Hippo pathway maybe